Opportunity to discuss our most recent published work on dynamic network analysis. I wish Yuri, who's my PhD student, or Willy, who's my undergraduate student, were able to attend the conference. However, both of them are international students, so they had problems with US visas coming back into the US after this, so they were not able to attend, especially Yuri, who's doing a Facebook internship, so he was not able to be away from the US for more than a week. Nonetheless, you're stuck with me, so hopefully you're not disappointed. So I'll tell you a little bit, motivate the problem, and then go into methodology evaluation, kind of key biological implications of our results, and give you some open questions, talk about some open questions that our methodology can be perhaps used to solve those problems. We study networks, and why? Because they're everywhere. Any real-world phenomena you can think of, or most of them at least, can be modeled with networks that model relationship, that capture relationships between nodes, such as biological networks. There we go. Such as biological networks. For example, in protein-protein interaction networks, nodes are proteins, and edges typically correspond to physical interactions between the proteins. So traditional static network research has been mostly static. For example, in computational biology domain, researchers have typically studied static network representation of the cell, even though cellular functioning, along with many other real-world phenomena, are dynamic. So why? Well, one key reason is simply we didn't have dynamic network data available. In social network domain in particular, with availability of online social network data that is dynamic, people have approached the topic of dynamic network analysis. In biological network domain, this is really still a taboo topic. So our key question motivating this talk is how to efficiently study temporal networks. And first type of approaches, which we will refer to as a static approach, basically means if you're given temporal network, set of nodes and edges that evolve over time, nodes can appear and disappear, edges can appear and disappear. Basically, you just take the dynamic data and aggregate into a single static network, where you keep all the nodes that appear in at least one of these, what we call snapshots of the temporal network, and you link two nodes if they are linked in at least k of the temporal snapshots. K being parameter you can vary. This is just a variation. There's lots of different versions of a static approach you can have, but general idea is take the network, aggregate, and the advantage is, well, you can now use this static network, which kind of captures some temporal information in it, to study. You can use established state-of-the-art methods for static network analysis to study this network. However, think about this network evolving one way versus the other. You will get the same aggregate network, so you will lose important temporal information if you just rely on an aggregate approach. Alternative way to do this is you observe each temporal snapshot as a static network, apply a given method, get some network property in each of them individually, separately, and then analyze time series of the results. How does that property of a network evolve over time? Whereas you definitely capture more temporal information compared to a static approach, and by the way, we call this approach static temporal approach. So whereas you do capture more information than previously, you still discard very important relationship that lies in between the different snapshots. So our motivation is, can we develop an approach that will directly compute property of a network while taking multiple snapshots into account simultaneously? And that's what we will refer to as a temporal approach. And of the current temporal approaches that kind of are related, or the most related to our work, are those based on motifs, network motifs in particular. So if you're not familiar with what these are, they're small networks, there is up to 2, 3, 4, 5 nodes at most or so, that are statistically significantly overrepresented in your data network compared to a random network with respect to some null model. And they're called by different names, but they are really just temporal motifs. However, all of these approaches, at least as implemented in the literature and in currently published papers, have serious drawbacks. First advantage is they definitely capture this inter-snapshot connectivity across time, but they do so by limiting the size of network structures, the motifs that they can consider. So they typically deal up to 3 nodes, or they limit the complexity of the structure, so they study linear paths or very simple structures, but not more complex ones. In many domains, in many different applications, especially in computational biology domain, it is not just relevant to characterize network property, basically property of the whole network, but also to zoom into local neighborhoods of nodes in order to characterize with a given approach their network position of individual nodes, and then link that node signature, network signature of a node, to its biological function. These approaches cannot do that, and we are, of course, aiming towards that goal. And the most serious issue with motifs, which we will try to address with graphlets, along with all the other issues, is motifs rely on a network null model. Motifs were introduced by Uriel Long's group in 2002 to 2004 range, and even immediately at that time, it has been recognized that you change the null model, you get different motifs from the same network. Okay, so that is in static case. In dynamic case, finding appropriate network null model is even a harder problem. So in 2004, so in parallel to network motifs, Natasha actually proposed the notion of graphlets that I will introduce in a moment, and then tell you how we generalize static graphlets into dynamic ones to allow for analysis of dynamic or temporal networks. I will try to keep this high level so that the majority of the audience could understand the methodology, especially if you're not directly involved in network research. For technical details, I refer you to the paper, and please feel free to talk to me after the talk. So graphlets are, if you want to be very formal, here's the definition, but graphlets are small graphs on up to n nodes, and in practical applications typically being 5-ish, that basically you're capturing all possible different structures on up to 5 nodes. So here I show you all possible structures on all possible graphlets on 2, 3, and 4 nodes. If you went to 5 nodes, there would be 21 of them, hence we left them out. But when we analyze networks, we actually go up to 5-node graphlets. Now, very related, these are static structures. So very related notion is that of dynamic graphlets. We just really, as simple as it sounds, the general idea is just to associate events, temporal information on the edges. So instead of dealing with a graph that consists of nodes and static edges, we're dealing with a graph that consists of nodes and temporal edges, or as we refer to them as events. So each of these events is associated with particular time at which it happens, as well as the duration of the particular event. So let's assume we have this triangle. I'm drawing an analogy with G2. So graphlets are numbered from G0 to G1, G2, and so forth. So we're focusing on the triangle. Let's say we have these kind of 3 events, Monday, Tuesday, Wednesday. Equivalently, because we are interested in relative order of the events rather than absolute time at which they happen, let's say that I'm dealing with events that happen on Friday, Saturday, and Sunday. And for simplicity, let's assume my network begins on Monday and ends at Sunday, so the whole week. The only difference in definition between, or I should say the only theoretical difference in the definition of static versus dynamic graphlets is the delta T connectedness. Each of these static structures is connected, meaning for every pair of nodes in the given graphlet, there is a path between them. The same definition applies to dynamic graphlets, except for every pair of nodes in a dynamic graphlet, there has to be a delta T time-respecting path between them, or in other words, in a given path, between every consecutive pair of events, consecutive in terms of time. In previous example, Monday precedes Tuesday, Tuesday precedes Wednesday, and here we have Friday precedes Saturday, and Saturday precedes Sunday. So for every consecutive pair of events on a given path, the later event has to start within delta T time after the previous event ends. And both of these different structures, because we are taking into account relative order of events rather than when they actually happen in real time, will correspond to the same dynamic graphlet, where this event will correspond to time slot k, time slot k plus 1, and time slot k plus 2. So we have a triangle that is now defined on, we have triangle that is now defined on 3 snapshots rather than in a single static snapshot. And now, if I just, to illustrate to the meaning of delta T on another example, if we have a triangle with Monday, Tuesday, and then Thursday, if my delta T is a single day like it was in previous examples, here, Monday and Tuesday are what we call delta T connected. Tuesday begins within 1 day of Monday ending. But Tuesday and Thursday are not, so we are no longer respecting the notion of delta T connectedness. So in order for this to be the same static graphlet I showed you before, delta T has to be at least 2 because of the 2 events, Tuesday and Thursday. If we generalize this, so let me just, sorry, before I jump into a formal representation of the graphlets, if this is my time evolving network, here is an example of what we call D2, dynamic graphlet 2, with 3 nodes and 2 edges, and the same structure, if you ignore the node labels, we would appear, for example, between D and C. Well, it wouldn't. Give me a second. Okay, perfect. So if I have between A and B, oh, they wouldn't. Never mind. There's multiple instances of this same particular graphlet, which I cannot see at the moment, so I'm going to skip it. But let me illustrate you the example of a triangle, which appears first edge of the triangle between B and C appears in T1, second one between C and A in snapshot 2, and the third one between B and A in third snapshot. Here are all possible graphlets. Here are all possible graphlets on up to 3 events. Unlike in static graphlets where we order them by the number of nodes, dynamic graphlets are defined with respect to both the number of nodes and the number of events. And here we group them based on the number of events they incorporate. And as you can see, multiple dynamic graphlets can be mapped to the same static graphlet. So by incorporating dynamic information into the process of what I will show you shortly, very quickly, characterizing the network structure, we are able to incorporate more of the information. We have detailed counts compared to what we are able to do with static graphlets. So several things we try to do without going into detail is once we have defined what dynamic graphlets are, how do you figure out the number of graphlets on n nodes and k events? We first devise recursive formula and then a closed form solution for this problem. And then the question is, how do you count graphlets, how many times each of the graphlets you want to count, how do you count them in a network? We have 2 ways to count this. One is, I will just say exhaustive enumeration, whereas the second one is more approximate counting. The output of our methodology is either how many times each of the graphlets of interest appears in the whole network, so we get graphlet frequency vector for the whole network, or how many times a node participates in each of the graphlets, in which case we get graphlet degree vector of a node. We do 2 different analyses. I will skip network classification in the interest of time and talk about node classification. For each node in the network, and we have different labels on nodes, either cancer or non-cancer, aging, non-aging, in social networks, manager versus employee versus CEO, and so forth. We compute, given a dynamic network, graphlet degree vector of each node, and kind of try to see if we use PCA, the principal component analysis, dimensionality, the reduction technique, can we correctly group in this 2-dimensional space nodes with the same label and separate nodes with different labels. We measure accuracy of doing this using established methods of area under precision recall, as well as area under ROC curve in biological network domain and social network domain. And we see that either dynamic or constrained dynamic, the difference being just how we count the graphlets, we basically see that they always improve upon both the comparable static and static temporal approach that are also based on graphlets. The only difference between these different approaches is how much of temporal information you're incorporating. I can comment on why the differences here are not even remotely as large as here. They're still statistically significant, and I would be happy to chat about this after the talk. What we did, and I already motivated why we study human aging, and I think it's straightforward in my highlight talk. So I want to comment, and this was the summary of the highlight talk that I mentioned. We are, from our previous study, we have aging-related temporal protein-protein interaction networks of human. So what we did previously is analyze each of the snapshots in isolation using a static temporal approach. Here we are trying to basically see if we use our dynamic graphlets as a basis of a temporal approach, can we improve predictions that we've done compared to our original approach. So if we again compare static graphlet-based approach, static temporal graphlet-based approach, and dynamic graphlet-based approaches, on how well we can, if we hide the knowledge about currently known aging-related knowledge, how well we can predict it. This is accuracy in terms of basically of all predictions that we make, how many of them are accurate. And you will see the superiority of both dynamic and constrained dynamic graphlets over the other approaches. Note that random approach performed quite well as well. And if you wonder why, that is because we have a lot more samples in non-aging related data than we do in aging related data. There's class imbalance. So if we try to correct for this, here's the actual more correct statistics, or at least less biased statistics that we believe in are more accurate. And again, now we see a drastic drop of both random and static approaches, whereas static and temporal still holds well, and dynamic is definitely superior again. So we make some novel aging-related predictions that are absent from the current ground truth data about aging, and we try to, because really there's nothing that we know about them, we try to find indirect evidence that they are biologically meaningful. So we compare functional overlap or disease overlap between our predictions and what is called sequence age in our study. Typically it's referred to as gen age. This is as trusted aging-related data as you can find out there. What I mean by functional or disease overlap, we find functions and diseases that are significantly enriched in our predictions and also among genes that are in this data set. And the overlap size is here, and p-value is indicated in parentheses. And you will see that only dynamic graphlets can find significant overlap, and the fact that our predictions perform similar processes as aging-related ground truth data is actually a very, very good sign. We tested a lot more cases than what I'm showing you here, and in 2/3 of the cases, dynamic graphlets were superior compared to both static and static temporal approaches, and we were comparable in 1/3 of the cases. There is a number of goal terms and disease ontology terms that were enriched in our predictions, but not in the ground truth data, and through literature search, we managed to link these processes and diseases to aging. I will conclude with a relatively short slide. This is the key point I'm trying to make. We have demonstrated using the notion of graphlets that the more temporal information we include, the better predictions we are going to get. Now, you can get as output the feature vector of the entire network or feature vector of individual nodes, and these two have been used with static graphlets in numerous computational and applied contexts, showing superiority over other network-based approaches. So by generalization, we are optimistic that dynamic graphlets, when applied to these problems in dynamic network research, will also lead to superior results. The code is available on this website. If you can't find it for some reason, please feel free to shoot me an email, and I'd be happy to point you to it. This is my group, and again, I want to acknowledge Yuri and Huili for all their hard work on this project, and this work was funded by NSF. Thank you very much. I don't know if we have any time for questions.
There is time for one question, one burning question, if anybody would like to ask. Yes. Can you
very quickly, in your comparison table, you have entries where it says NA. What does that mean?
That means there was a single gene in the there was a single goal term or dot term in the overlap, and you cannot measure statistical significance with only one gene because the formula for hypergeometric test does not allow that. Yeah. I'm happy to talk about any questions you might have after the talk. Thank you very much.